TCA cycle, which is also known as Krebs cycle or citric acid cycle. It is the final common pathway for the carbohydrate, fats and protein for their breakdown with the final production of CO2 and H2O. It takes place in the mitochondrial matrix of the cell, and it is amphibolic in nature, that is, it have both catabolic as well as anabolic role. In catabolic role, the acetyl coenzyme A produced from the metabolism of carbohydrate, lipid and protein are oxidized in this cycle to produce CO2, H2O and energy, that is ATP. Now moving on to anabolic role, that is synthetic role, and the intermediates of TCA cycle are here utilized for the synthesis of various other compounds. For example, for the synthesis of heme, succinyl coenzyme A is used for the synthesis of porphyrin and hemoglobin, and synthesis of non-essential amino acid purines and pyrimidines. Oxaloacetate and alpha-ketoglutarate serve as a precursor for the synthesis of aspartate and glutamate respectively, which in turn are required for the synthesis of non-essential amino acid as well as purines and pyrimidines. Again, for the synthesis of fatty acid, mitochondrial citrate is transported to the cytosol where it is cleaved to provide acetyl coenzyme A for the biosynthesis of fatty acids, sterols, etc. If we are to take only the intermediates of TCA cycle that help in the further biosynthesis of other molecules, the cycle will look like this. So, as already explained, the citrate of the cycle can be utilized for the production of fatty acids and sterol. Alpha-ketoglutarate help in the production of glutamate, which in turn helps in the production of amino acids and purine. Succinyl coenzyme A help in the production of porphyrin and heme. Oxaloacetate help in the production of aspartate, which in turn helps in the production of other amino acid as well as purines and pyrimidine. Moving on to the reactions of TCA cycle. The first step is the connecting link between glycolysis and TCA cycle. Because we get pyruvate from glycolysis, the reaction is catalyzed by pyruvate dehydrogenase. Here, pyruvate undergo oxidative decarboxylation reaction to yield acetyl group, where NAD is reduced to NADH along with the release of CO2. The proper TCA cycle starts catalyzed by the enzyme citrate synthase, where 2 carbon containing acetyl coenzyme A condense with 4 carbon containing oxaloacetate to form 6 carbon containing citrate. Here, acetyl group of acetyl coenzyme A is transferred to oxaloacetate, where H2O is required to hydrolyze high energy bond linkage of acetyl group and coenzyme A. This energy release is utilized for the condensation of citrate. The citric acid is then converted to isocitrate with the intermediate cis-acotinase. Here, both reaction is catalyzed by acotinase, same enzyme, along with the ferrous ion. The initial reaction is dehydration reaction followed by the hydration. Isocitrate then undergoes series of reaction to form oxalosuccinate to alpha-ketoglutarate. Here, both the reactions is catalyzed by the same enzyme, isocitrate dehydrogenase. Initially, isocitrate undergoes dehydrogenation reaction catalyzed by enzyme isocitrate dehydrogenase, where NAD is reduced to NADH. In second step, the oxalosuccinate undergoes oxidative decarboxylation reaction to form alpha-ketoglutarate. Reaction catalyzed by the similar enzyme. Alpha-ketoglutarate then undergoes oxidative decarboxylation reaction to give succinyl coenzyme A. It comprises of various coenzymes and prosthetic groups such as magnesium ion, NAD, CoASH, lipoic acid, etc. This succinyl coenzyme A is further converted to succinate in the presence of enzyme succinate thiokinase. Here, GDP is converted into GTP, and this GTP is further converted into ATP by the enzyme nucleoside diphosphokinases. Succinate is oxidized to fumarate by the presence of enzyme succinate dehydrogenase. The reaction is dehydrogenation reaction, and FAD is the hydrogen acceptor group. Fumarate is then converted to malate in the presence of enzyme fumarase along with the presence of H2O. The enzyme is fumarase, and finally malate is converted into oxaloacetate in the presence of enzyme malate dehydrogenase. Here NAD is reduced to NADH with the final product of the cycle, that is oxaloacetate. And if you remember, the oxaloacetate is the same product which helped to initiate the TCA cycle in the very first step, where it condenses with the acetyl coenzyme A to form citrate. So the cycle continues where oxaloacetate is reutilized in the cycle, and in this way the TCA cycle is completed. So if you are to take only the intermediate substrate from the cycle, then the cycle will look like this. Initially, there is formation of acetyl coenzyme A, which condenses with oxaloacetate to form citrate, which is converted to cis-acotinase, then to isocitrate, again to oxalosuccinate, that is converted to alpha-ketoglutarate and succinyl coenzyme A, then succinate, fumarate, malate, and again reconversion of oxaloacetate, which again condenses with acetyl coenzyme A, and the cycle continues. Now moving on towards the energetics of TCA cycle. If the TCA cycle starts from glycolysis, then there is 6 ATP production on the first step, where there is conversion of pyruvate to acetyl coenzyme A. Then again 6 ATP at the step where there is conversion of isocitrate to oxalosuccinate. Then again 6 ATP from the conversion of alpha-ketoglutarate to succinyl coenzyme A, and from succinyl coenzyme A to succinate there is production of 2 ATP, and from succinate to fumarate there is production of 4 ATP, and finally from malate to oxaloacetate there is again production of 6 ATP. So overall there is 30 ATP production if the cycle initiates from or continues from the glycolysis. So this is all about TCA cycle, its introduction, amphibolic nature, pathway, as well as energetics. Thank you.